Well, good morning. My name is Ryan Ashley. I'm a professor in the Department of Animal and Range Sciences here at NMSU. My area of expertise is reproductive physiology. So I'm actually super excited to be a part of this project. I am just now joining. I was contacted by Yan Yan and Dr. Zhu and Pei, and we started talking about just looking at some reproductive effects and any type of reproductive disorders or dysfunction, anything that associated with repro in terms of this treated produced water. And so I got really excited about this, I think, and so I started delving into the literature, and surprisingly, there's really a dearth of knowledge, right, on treated produced water and any effects on reproduction. So as a reproductive physiologist, I was like, oh my gosh, this is like a wide open field, like, here we go, let's go. And so we have come up with at least a general kind of pilot study to kind of start going through and trying to answer some of these reproductive questions. Let me make sure I don't screw this up. Okay. So this is kind of what we're proposing, so kind of following along with my colleagues, Jean and Yan Yan. So we're going to continue using the same type of mouse model, so this Swiss outbred mouse, and like Dr. Zhu elaborated on, one of the benefits of this is that these mice, since they're not inbred, they're going to be much more similar to humans than like an inbred mouse line, like a C57 Black 6 or something. But overall, what we're wanting to do is to use these mice and trying to keep it simple to start with, and just having essentially 3 treatment groups. And this is where nothing is really set in stone. I need to still work with my collaborators on this. But just starting out simple, having like a control group, a raw produced water, and a treated produced water group. And then overall, our objectives really for this mouse study on the reproductive side is we want to know and evaluate, the perinatal and early life exposure. So if these mice are getting exposed to raw produced water or treated produced water during these time periods, is it having an effect on reproductive development? Also, we want to assess, I think both Yan Yan and John are showing some really interesting, at least from my perspective, from a reproductive physiologist, seeing this kind of sexual dimorphism is really intriguing. And wanting to see, is there a difference on how males and females behave, if you will, how their body reacts to this treated produced water. And so another objective of ours is we want to look at these sex-specific differences in the developmental and reproductive events associated with our treatments. And then lastly, really looking at, wanting to investigate some molecular and hormonal markers of early reproductive axis activation and disruption, and just see really, what effect is there any effect of treated produced water on reproduction in a mammalian system? Our overall design to start with, we're proposing to essentially take mice, and they will have like a pre-exposure time, so 10 weeks in which they're going to be exposed to one of these 3 treatments, the control, raw produced water, or the treated produced water. And so that will be for 10 weeks, and then we will start mating. And so we'll do a one-to-one pairing, a male and a female. They will stay on their same treatments throughout the duration of the study. And so we'll have, again, the control, raw, or treated produced water. And so these mice will stay on that study from breeding, so mating, through gestation, and through lactation. And so one thing that we're going to do is then, so for example, at weaning, so it'll be about 3 weeks of age in the mouse, we'll take a subset of those pups, and then also we're going to have a group that goes further on till right about the time that they would reach puberty, and we'll also harvest a group of pups or mice at that time point. And so in all these studies in which we collect tissues from these mice, we're going to be looking at some key things. One, just the developmental and some physical milestones, looking at body weights, right, of all the internal organs, different things like that. And though I didn't really put it up, well, I'll come back to that in a second. But that's going to be some of the key things, similar to what Dr. Jean Zhu was looking at, just trying to see if we're seeing any changes in just weights. The nice thing here, too, is, we'll collect and keep all these tissues, and so we can come back and do different molecular analysis and look at things associated with inflammation. We can look at whatever we want to in terms of the gene expression and histology approaches. Another key thing we want to look at is, are we affecting the onset of puberty? Are we changing the puberty onset for males or females depending on treatment? Then, of course, one of the things I'm really excited about is just the reproductive parameters. I didn't necessarily put it up here, but some key things we'll look at, right? I was laughing at myself as I was looking at these slides last night. As a reproductive physiologist, all these cool things I want to do, I didn't even put up on the slide. But some of the key things we'll look at is just, are we affecting breeding rates? Is there a lower percentage of conception, right, based on treatment? Is there a difference in pup numbers? Is there a difference on, conception rates? And so there's a lot of key things we can just look at just from the breeding and just how many pups we get and some of those developmental milestones. But also what we'll do again at both the pups that are at weaning and those that are reaching puberty, we'll look at their testes and ovaries, and so we can look at just development of those, the maturation. We can start to look at different molecular analysis in terms of, are they making the correct steroids, are any of these pathways being manipulated? And so there's a lot that we can look at just looking at their primary reproductive organs, which is the testes and the ovaries. And then granted, though it is kind of hard to get a lot of blood from a mouse, we will use what we can to try to look at some hormonal and other molecular analyses to try to see if they're having any reproductive effects associated with treated produced water. Hopefully there will be none. And so that's, very general what we're wanting to do to kind of start looking at some of the reproduction effects. Some of the key questions that we're hoping to answer with beginning these studies is one, are there developmental or reproductive toxicities associated with treated or raw produced water? Does early life exposure to treated produced water, does it affect the onset of puberty? And again, coming back to some of my colleagues' interesting data, are there sex-specific differences? Is there a sexual dimorphism here? Is there something that's different between males and females and how they respond to treated produced water or raw produced water? And then just as important, we want to know what effects does this happen just on hormone levels and hormone regulation and production, which is key to just overall reproduction in the mammal. And then similar to what Yang was talking about, we'll also look at a number of analyses to look at inflammation and oxidative stress in these tissues to give us a better idea of what's happening from a mammalian side when they're exposed to these treatments. And again, going through this and just reading up in the literature, I was getting really excited because there are so many studies that can be done. So we're really trying to start out with some basic, simple studies to give us an idea of what is happening. A lot of my expertise really is in placental biology. And so one of the first things I was thinking about was like, oh man, it'd be really cool to see, are we affecting like implantation rates? Are we affecting how the embryo attaches to the uterus? Are we affecting how the placenta forms and functions? So I think these initial studies will really give us a firm foundation, which there can be a plethora of other studies that are going to be tangential to this that we can also conduct that will really give us a greater understanding of what might be happening from a reproductive standpoint. So again, we haven't even started any of these studies. We are in the process. We've got an IACUC protocol in and submitted. So as soon as we get approval, we're hoping to start these studies hopefully this summer. And so like Dr. Zhu talked about, these studies will take place in our new biomedical research facility, which I know some of you are going to get a tour of later this afternoon. And so this is, we're excited about this. It's a great facility, and we've got a lot of great students that are going to help work on this project as well. So anyways, I just want to thank you. This is kind of my first introduction to this consortium. I'm really impressed with already the quality of work that's taken place here, and I'm very appreciative to be a part of this and look forward to working with you guys for many years, hopefully. Thank you, Pei.
Yeah, thank you, Ryan, Yan, and Dr. Xu for the excellent presentations.